Hi, good evening. Thank you for this invitation. Tonight, I'm going to talk about wound healing and the treatment with new technology. Anti-aging is equal health. So we are working on improving health for the cells in general. And our investigations and technology has different complex treatment. It's very complex, all this research around aging and anti-aging. It has been going for a long time. And we focus especially on molecular bases with this technology. And there are two factors that we take in account, and that is the biological circadian clocks and the cellular communication delivered in a specific order defined by time. Our cells have a circadian clock, and it's essential, this cycle, for the immune control mechanism. Every single cell in our body is controlled by its own circadian clock. It helps every cell figure out when to use energy, when to rest, when to repair DNA, or to replicate DNA. The Nobel Prize in Physiology or Medicine 2017 was awarded for Geoffrey Hall, Michael Rauschbach, and Michael Young for his discovery of molecular mechanism controlling the circadian rhythm. Cells are governed by their biological clocks in order for optimum communication to take place between artificial intelligence, blueprint signals, and naturally occurring biology signals. The AI signals must be delivered within predefined variable times that maps the time schedule of biological signals. Therefore, the iRevive is designed on the basis of a matrix of signals delivered within a time matrix. I think we are going fine. This is part of our experiment model of a protein and how this protein move into the direction of changes through a resonant and signaling of the communication. And you can see that the resonant energy stabilizes the protein and connect the two parts in much closer position. And that gives you an aspect of how this signaling and works and how the energy and the resonance energy is transferred. What are the 5 basic mechanisms of signaling that we use and that's important for us? The first are the blueprints that resonate on in it's very funny. The blueprints signals resonate on in synchronic way with biological signals. The ultra-low properties energies based on a mathematical proof, electron-gated ion channels that we have seen in 2008, where electrons control and amplify ion channels, is where this actually works on a molecular level. Signals are emitted in variable discrete times. In all of time, we talk about circadian rhythm timetable. And multiple resonant signals emitted simultaneously. And a mega antioxidant effect by the electrons carrying the signals into the system. Five basic mechanisms of signaling. Blueprint signals resonate in a synchronized way with biological signals. Ultra-low properties energies based on a mathematical proof, electron-gated ion channels, where electron control and amplify ion channels. And in point of interest, signals are emitted in variable discrete times, circadian rhythm timetable, and multiple resonant signals emitted simultaneously. Mega antioxidant effect by the electrons carrying the signals into the system. So signals are very complex. What we talk about signals is signals are, as you see, waves of sound, and they are different waves. They come in different complexity, and for every cellular function and mechanism and signal, there's a complex signal or wave that resonate between cells. So here you can see that as I said, resonate means noise, of course, and that can be measurable. And as you see that most of the time we use microvolts in order to get much more accurate signals and much penetrate to the cells and much specific function and pattern in these blueprints that is that control the cell signals. As we advance, we talk about DNA, and it's one of the previous slides where we talk about DNA transcript and DNA division and DNA repair. And in all this cell injury, we will have a natural signal that will reconstruct and transcript a DNA. And that occurs, of course, in an unfolding manner of the DNA's chain, and as we know, how it goes and transcripts itself. So this is one of the proteins that has been studied in using a plasmon surface resonance. It's one of the other technology that has been used to unfold protein and read these signals much more better in order to get this communication. So signaling to repair DNA, of course, this is what we are doing with this technology, trying to repair the DNA in open wounds, in wound healing. And in this research, we focus on the investigation of biosignals and how timing affects their meaning within the biological network. One of the diseases that we have studied and has been studied for a long period of time in dermatologists and is psoriasis. Psoriasis is one of the diseases that has like his own memory, his own cell. That means he he multiplies in a very rapidly way, and it has a lot of inflammatory process. Because of this in the independent way, the psoriatic cells multiplies themselves. You can see that at the beginning, you can see there's acanthosis, very important acanthosis in psoriasis patients, and you can see this, all these markers that is the pattern of the inflammation of these cells. Of course, this is telling me that the time, the circadian time of these cells are interrupted, and they are rearranging themselves in an independent way. And that's why it's very interesting to compare this kind of disease and study it to understand what we're talking about, this circadian clock of these cells in this disease. So follow this, we can see treatments that we have been done in this kind of patient who had psoriasis, and which we can see there's a total recovery of the inflammatory skin problem. Another activity that can be measured in inflammatory pathway is the effect of viruses that activated transcription factors in proteins pathways, and they do extracellular cellular regulated kinase, activate transcription factors to c-Jun and terminal kinase pathway. And what we can see here is that the treatment of these kind of inflammatory infections always also can be regulated by signaling pathways, and we can target these segments to enhance the cell cycle. So as the treatment goes on, this you can see a patient that has been treated with this technology, and you can see the technology has restored the inflammatory process of the skin. It has restored the inflammatory process of the nerves, because this herpes produce an inflammatory injury to the peripheral nerve of the skin, and you can see on the face of the facial nerve. And this repair of the DNA is not only in the sense of retranscription only or repair, it's also taking into account the timing of the cell, which will give you his own repair cycle, or his own repair cycle in order. Let's say it like that. So as you can see, the results are very, very quick, and it works on any level of the skin structure. So it can be also, like I said, proteins unfolding, folding and regulating signals pathways. And it's very important because, of course, experimentally you can experiment on protein regulation, but then you can do it on every cell of the human body. So anti-aging is about going back in time. Why can't we go back in time? the greater the amount of complexity, the greater the difficulty of going back in time, of course. Why would we want healing? And this is also part of going back in time, because when we do an injury, the cell repair himself, and there's multiplication of DNA, and the wound closure spontaneously. What happened there? What we want to do here, and what we want to achieve? It has been studied, the mechanism of coagulation and the angiogenesis in wound repair. And also, after this angiogenesis, we can see there's there are more enhancement of the collagen or collagen repair. And that's what we want in wound healing. But what else do we want? We want something more. We want actually that not only the wound healing is collagen and angiogenesis, but also we want this angiogenesis and this collagen repairs with the other structures they are in the skin. Why? Because the skin does have many other cellular organisms that need for his complete healing, like nerves ending, like elastin, like hair shafts, etc. And we know, and like glands, sweat glands, fat glands, all the structure of the skin, because the skin has many structures that are functional for his integrity. So here you can see more patients with wound recovery after treatment with eye repair, and you can see the skin has been recovered in its whole structure. Also, we have treated many patients with diabetic foot, and one of the goals are not only the wound has been closed in shorter time, but also the neuropathic problems that you can see in diabetic neuropathy is very, very important because of the treatment of this technology on the nerves ending, removing or enhancing the function of the nerve and reducing the inflammation around the nerve ending that is caused by inflammation and, of course, glucose levels. Also, you can see how this skin repairs has been very successful in his total structure. And as I said, all the cells of the skin has been enhanced and repaired with this technology. Here you can see a patient with striae and also have very good results with this. And this is another one I would like to show you here in this picture, because this wound is in the healing process, and when this healing process, like in the first days of healing, repairing of the cells, is where this technology can repair much more structure of cells of the skin than skin that has already been healed or scars that have been there for many years, because there's no more inflammation there, and there's no repairing factor, there's no more repairing cells ongoing. So here you can see this scar that is fading away on this side. So in conclusion, wound healing has been studied only by his endogenesis process. With technologies of with the technology of resonance signaling, we are not only restoring angiogenesis, but also resonate through signaling with the collagen repair of the skin, the intrinsic nerve system of the skin, and inflammatory mediators. This stabilized wound repair of the skin and all the structure, which, as you can see in the results, a complete recovery of all the skin layers, inclusive the hair shaft. Thank you for your attention. Thank you for the invitation again. Hope you enjoyed the lecture. Thank you. And Gumi, you can stop sharing and we have to end the lecture.